Well, thank you very much. I'm excited to share some of our work from the Rangamani Lab at UCSD with all of you here today. I'm going to talk about one particular example of looking at realistic cell geometries and how that matters for cell signaling. So we all can probably appreciate that cell signaling is quite complicated in many ways, right? For one thing, there's a lot of different reactions that are occurring between many different molecular species within the cell. From kind of the physics standpoint, there's this very interesting feature of having reactions that are occurring between different volumes within the cell and surfaces on the cell, usually consisting of membranes like the plasma membrane or membranes of organelles. And this leads to these interesting mixed dimensional systems of partial differential equations that can be difficult to solve, especially when we consider that cells themselves have quite complicated geometries, like we just saw with the microtubules, right? There's very specific arrangements of organelles within the cell. This is from a volume electron microscopy data set. And so to actually consider the cell in its full detail would require a lot of computational effort, right? And so we've been making some strides towards doing this, towards running simulations within realistic cell geometries. And in the example that I'll show today, we're focused on the case of mechanotransduction. So mechanotransduction, kind of in a typical sense, we might think about cells responding to the stiffness of a substrate that they're on, right? So, but that's just one aspect of cell mechanotransduction. They will also, in general, respond to the topography of the surface that they're embedded within. So cells in tissue might experience very complicated fibrous networks. For instance, this is what some example kind of collagen gels might look like, and an example of a cell embedded in a collagen gel, as we've already seen today. And so you can appreciate that the cell is going to respond to a lot of different cues here and would need to be able to adapt to actually migrate through such spaces. So we consider kind of a simplification of this general format where now we have the cell responding to these specific structures on a substrate, which are these vertical nanopillar structures. And as you can appreciate in this image, these nanopillars will induce very large deformations of the plasma membrane as well as the nucleus of the cell. So we want to represent this in our simulations in order to answer the question of how these nanopillar substrates might influence mechanotransduction signaling by changing curvature of the plasma membrane as well as deformation of the nucleus. So to do this, we used our recently developed software framework known as Spatial Modeling Algorithms for Reactions and Transport, which conveniently spells out SMART. And one example that we looked at in just this original paper that we put out with our framework is this mechanotransduction network, which many of you may be familiar with a little bit, where we start with some sort of event at these integrin binding regions at the cell membrane, which leads to phosphorylation of focal adhesion kinase and eventually activation of the cytoskeleton, which will then free up YAP-TAZ within the cytosol and allow these molecules to move into the nucleus. The reason that we care about this is that YAP and TAZ will bind to TEAD molecules within the nucleus and induce transcriptional changes within the cell. So in general, we would expect more YAP-TAZ to make it into the nucleus on a very stiff substrate. We ran some tests in our framework for different cell shapes, in this case under the assumption that they're adhering to very specific micropatterns, which had actually been tested experimentally previously. So this is for like a star pattern and then for a rectangle pattern. And you can kind of appreciate that the cytoskeleton gets preferentially activated at regions of high curvature along the perimeter of the cell contact line. And this leads to slightly higher level of YAP-TAZ making it into the nucleus for this cell. So we wanted to adapt this to cells on a nanopillar substrate, right? So in addition to changing the geometry itself, we also made some additional assumptions based on what's been shown in the literature. So one effect that we know is going on these nanopillar substrates is that we have preferential actin assembly on regions of positive inward membrane curvature around these nanopillars. And this is NWASP mediated. So we kind of phenomenologically inserted this into our model And we also put in this curvature-dependent endocytosis, which leads to a reduction in focal adhesion kinase phosphorylation at these curved regions. And this is due to endocytosis, specifically of beta-1 integrins in this case. So putting all these assumptions and the geometry together, we ran our simulations within some sample cell geometries. So in this first case, you'll see for early time points in our simulation, we have this assembly of actin around these nanopillar structures, which is exactly what we put into the model, so kind of a sanity check. That's what we expect to see. Now I'm going to play over a longer time period and a higher range of concentrations of actin. Full play. Oh, I was impatient. Did I stop it? There we go. Okay, so in this case, you can see actin assembling in our continuum assumption just throughout the cell, leading to movement of YAPTAZ into the nucleus. If we compare this to a cell on a flat substrate, then we could see that there's comparatively more assembly of F-actin and more movement of YAPTAZ into the nucleus. Well, it's a little hard to appreciate with this color scheme. But the slight increase in YAPTAZ is essentially due to the reduction in FAC phosphorylation here. And by tuning this FAC phosphorylation inhibition, we were able to show good agreement with previous experiments which tested cell response to different nanopillar substrates as shown here. So that's good. We can kind of recapitulate some of these effects that have previously been shown. Now, an interesting thing that we haven't talked about yet is what happens to the nucleus deforming on these substrates, which hopefully you could appreciate from the first image that I was showing. So our collaborators in the Jawhead lab at UCSD Nanoengineering measured nuclear deformations in U2OS cells on nanopillar substrates. They showed that you get very large amounts of indentation of the nucleus, several microns, on these substrates. And even more astonishingly, they found that cells with deformed nuclei on the nanopillars tended to exhibit these nuclear envelope rupture, which you can see evidenced here by Ku80, which is normally confined to the nucleus in cells on a flat substrate, into the cytosol of the cells on the nanopillars. So the nanopillars are actually rupturing the nuclei of these cells. So naturally, we kind of wondered what happened, how does this affect YAPTAZ transport due to both stretch of the nuclear envelope leading to enhanced entry through the nuclear pore complex, as well as nuclear envelope rupture. So we introduced nuclear indentations into our geometries, and we were able to compute associated stretches of the nuclear envelope. And so specifically what I'll show here is the effects of nuclear envelope rupture predicted by our model. So to induce nuclear envelope rupture, all we did in this case was prescribe an enhanced entry rate at this central region of the cell at a specific time. And so if we play this video, what we can see is a very rapid entry of YAPTAZ after the induction of nuclear envelope rupture, which happens kind of fast, but we can see it again. So this is somewhat unsurprising, but it depends on the assumptions you make about as far as like what's the preferential entry rate versus exit rate. But in the end, our model was able to predict an enhanced amounts of YAPTAZ within the nucleus here. And we were curious if this would be the same thing you see in experiments, because they had only shown that these rupture events occur, but hadn't measured YAPTAZ. And what we see here is when they look at YAP within these same cells that had exhibited nuclear envelope rupture, they have an increased amount of YAP within the nucleus in line with our simulations. So in summary, we investigated these multiple ways in which nanopillar substrates regulate mechanotransduction via plasma membrane curvature, either through local actin polymerization or through integrant endocytosis leading to suppression of focal adhesion kinase phosphorylation, or through nuclear envelope stretch and rupture, enhancing the entry of YAPTAZ into the nucleus. And this would have very distinct effects on cells, leading to changes in gene expression. Now, I want to leave off with kind of the next directions in terms of this project. What you may have noticed is so far I was just prescribing these geometries, for instance, of the deformed nuclei. So one thing we want to do is to understand exactly how you get to that conformation. So for that, we can actually run mechanical simulations of nuclear envelope deformation. For instance, by applying a force on top of the nucleus and pushing it down onto the nanopillars. If we treat the nuclear envelope as a hyperelastic sheet, then we can predict the amount of stretch of the nuclear envelope over different regions, as shown here. So this is just one preliminary result that we had. The end goal is to find these regions where we have potential sites of rupture and then couple our transport model to this mechanical model of nuclear envelope deformation to actually close the loop and be able to kind of have these mechanochemical predictions on the cell scale. So with that, I want to acknowledge my PI, Professor Rangamani, as well as our fellow lab members and collaborators in the Jahed Lab in nanoengineering, as well as our collaborators at Simula who really help develop our software, and also our funding sources, and thanks to all of you for listening.
Okay, we have time for a few questions. There's one in the back.
Yeah, I think that's a very good point. The main thing that I would worry about is there's two membranes, and then there's also the lamins underneath those membranes. So those are kind of distinct layers, right, in some way. So effectively, we're kind of lumping all those things together, including the nuclear pore complex, as you're mentioning. I think that's something we'd have to investigate. I would start to think about the length scale that we're looking at, right, in terms of the predictions that we're making. But that's a good point.
Can you think about perturbations that will basically cancel each other? So for example, if you press on the nucleus, but at the same time the cell is sitting on nanopillars, and so one increases YAP signaling, the other one decreases it, and have people try these perturbations experimentally?
Yeah. So I think you could essentially do that. It's something we could predict in our model very easily, but I think it would be hard to tune the experiment, see what exactly cancel those things out. But I think if we kind of sort through the experiments in the literature, we would be able to find effectively that they did measure this already in some of that initial data that I was showing.
But by kind of like playing around with these two perturbations, can you measure the relative contributions of nuclear deformation versus cytoskeletal deformations in YAP signaling?
Yeah, so you definitely can do that in theory, right? Why is that not going? Yeah, so on average, they saw like a reduction in YAPTAZ on these nanopillar substrates, right, which I was showing initially. And so, but then at the same time, you have select cells with increased YAPTAZ due to nuclear envelope rupture. So I think that in reality, there's heterogeneity across the cell population, and that's part of the reason that some of this gets washed out in the data. But I think we definitely could do more work to actually calibrate to the experimental data and really constrain some of these parameters that are regulating the YAPTAZ transport. Yeah.
And so one more thing. So do you think these two effects are additive, or do you think we'll have some nonlinear interaction?
I mean, I would start by assuming that they're more or less independent of one another, although the local actin assembly would affect the deformation of the nucleus, for one thing, I think. So that's something we'd have to consider in our mechanical model, so that the actin that's actually between the plasma membrane and the nuclear envelope could actually induce some sort of nuclear deformations, for instance.
Okay, thank you so much for your talk. Let's thank our speaker one more time.